Biotechnology. Now, principles and processes from time immemorable, we have been using microorganisms for food processing, making curd, making bread, making wine, and so many things. But now we have more modified biotechnology by using genetically modified organisms, by producing the enzymes on large scale so that we are able to use it. Now, test tube baby. The sperm and the ova are put into the test tube and they are fertilized outside the baby. That is also included in the biotechnology, is by in vitro fertilization. Now, synthesizing a gene, a new gene, synthesizing by combining many genes, cutting it and combining that makes a new gene. So, synthesizing a new gene is biotechnology. Using that synthesized gene is a biotechnology. Now, there might be some genetic defect in the human or other organism which can be removed by biotechnology. The gene can be corrected by biotechnology because it's very advanced now. Now, developing a DNA vaccine, developing a new DNA and introducing it into the organism so that it develops the immunity, that is DNA vaccine, that is also very advanced nowadays. Now, European Federation of Biotechnology, they have included the traditional and the modern molecular biotechnology, and they have given a definition which includes both the integration of natural science and organisms, cells, parts thereof, and molecular analogues. Analogues means counterpart. Whatever molecule is there, their counterpart is included for the final product, and the service which is given by making a new product, new DNA, new gene, and making it beneficial for us is included in biotechnology. Now, the principles of biotechnology. Now, these principles of biotechnology are based on genetic engineering and bioprocess engineering. Now, what is genetic engineering? You have to change the genetic material, that is RNA and DNA. So, alter the genetic material and put into the host organism so that the phenotype is changed. You have to change the genotype and therefore the phenotype changes. So, that is genetic engineering. Now, bioprocess engineering is very important because whatever you have created, that has to be multiplied without the undesirable microorganism growing. So, you have to maintain a sterile condition. So, sterile ambience is must. Desired microbes or desired cell has to be grown in the lab, whether you want it for antibiotic, vaccine, or enzyme. So, the principles of biotechnology are based on genetic engineering and bioprocess engineering. Now, two types of reproduction takes place, asexual and sexual reproduction. In asexual reproduction, the genetic material goes on as it is, there is no variation. But in sexual reproduction, combination of genetic material takes place and therefore there is variation. Variation in the characters which help our survival. Now, humans have tried to use this character, so they have used animals by hybridization to create new characters. Now, these characters, whatever the desirable characters, along with that, the undesirable characters also come into the phenotype. Now, what we have done, what the genetic scientists have done, they have created the desirable character genes, transferred that desirable character genes into the organism and created a phenotype which does not have undesirable character. So this genetic engineering has helped us to create a recombinant DNA, a new DNA which we have created by combining two types of DNA and putting the desirable DNA into the chromosomes to have the desirable characters in the organism. So we have created recombinant DNA. We have done gene cloning, that is the desirable DNA, which is now a recombinant DNA, is made into gene. The gene has cloned and then we transfer the gene into the organism for the phenotypic characters. Now supposing you take a DNA and introduce into a cell, it is not going to multiply. It is just going to die in the new cell, in the new environment. But if you introduce that DNA at the origin of replication, you introduce that DNA at the origin of replication, is the sequence of the DNA in the chromosome where the origin of replication starts. When you introduce that DNA at the origin of replication, then the multiplication of that recombinant DNA takes place. It gets mixed with the genome. It gets attached to the genome and a new genome is formed. So it gets integrated with the new genome only when you introduce it at the origin of replication. So now this DNA will get integrated in the genome of the recipient and then multiply and then inherited with the host DNA. So the DNA is cloned. The cloning of the DNA takes place, or it is the multiplication of the template of DNA takes place. So that is genetic engineering. Now the first recombinant DNA molecule, how the first combinant, how the DNA was recombined to form a new DNA with the desired qualities. Now there were two scientists. One was Herbert Boyer and Stanley Cohen. These two people were working differently. Now Herbert was working on this restrictive enzymes. Restrictive enzymes are the molecular scissors. They can cut the DNA at the specific site to get the desired DNA. So he knew where to cut from and get the desired DNA. And Stanley, what he knew was how to take out the plasmid and transfer it into the desired cell. So when they came together in 1972, what they did is they took out these genes of antibiotic resistance from Salmonella typhimurium. This is the bacteria, Salmonella typhimurium. They took out the antibiotic resistance gene. The genes which encoded for antibiotic resistance. That means if I am a bacteria, antibiotic is poured on me, then I die. If I am resistant, then I don't die by this antibiotic. So this bacteria, they are their antibiotic resistance due to the plasmid which was present, which had DNA for antibiotic resistance. So these two scientists, what they did, Now this is the plasmid. This has got the DNA which will synthesize the antibiotic resistance chemical. Now this was cut with the help of molecular scissor. Molecular scissor gave a cut, and after cutting they were placed on the other plasmid. This is the host plasmid. Here the cutting is done. Once it is cut, it has got sticky ends. It is sticky ends. A cut is given to this by annealing, by giving it temperature, making the DNA single stranded, and cutting here with the restrictive enzymes. Now here, once the cut is given, this part, that is the antibiotic resistance, is fixed in this plasmid. Now after fixing the plasmid in this plasmid, we have recombinant plasmid DNA. So this DNA has got the new character. Plasmids are the structure present in the bacteria other than their genetic material, which are round DNA floating in it, has got extra characters for their survival. So here now this plasmid has become a recombinant plasmid DNA. That is, it has got, previously that plasmid did not have antibiotic resistance, but with this transfer of this DNA, here space is created by annealing, by removing some genes. Here by annealing is making the DNA single stranded, and then removing some sequence to make the space for this genetic material, which has got sticky ends, and these ends are also sticky. So this sticky ends will fix here, and it will become a structure like this. This is the point of attachment. So this is a new plasmid, which is a recombinant plasmid DNA. Once this DNA with the desired character of antibiotic resistance now will be put in the desired cells. Now the desired cells were Escherichia coli, which was very much similar to Salmonella typhimurium. Escherichia coli. Once the plasmid enters the Escherichia coli, now this plasmid acts as a vector in making it enter into Escherichia coli. Vectors are what? Like mosquitoes, they bite us. That is, they are acting as vector. They bring the microorganism from one human to another and just transfer the microorganism. So they are vector. Same way, plasmids are just transferring the characters into another organism. So they are, plasmids are vector. Now this plasmid has entered Escherichia coli, and it is circular plasmid. There are many more plasmid also, but this is the desired DNA plasmid which is floating. Once the Escherichia coli, they multiply, this plasmid with this desired character of antibiotic resistance also started multiplying. So this biomolecule goes on multiplying, and then we culture Escherichia coli. More and more antibiotic molecule will be produced. That has to be separated and then put into the bottle. You have the chemical, that is antibiotic resistance. Now this was the initial stages of discovery, but the discovery can be done with any type of genes like growth hormone, insulin, and many more. So first was forming a recombinant DNA molecule. Here the scientists Herbert and Stanley, they did it with the antibiotic resistant gene encoding antibiotic resistant biomolecule. that was in Salmonella typhi. They took out the genetic material, they took out the DNA by restrictive enzymes. They gave a cut and it led to sticky ends. They removed some genes by annealing, and then there was sticky ends here, and this part was fixed here, that is antibiotic resistant genes, DNA, recombinant DNA was formed. So now we have recombinant DNA in plasmid. Plasmid is just a vector and it was transferred in Escherichia coli. Escherichia coli, by polymerase enzyme, it multiplied the plasmid. Polymerase enzyme was present in Escherichia coli. So it led to multiplication of this recombinant plasmid DNA. So more DNA, more multiplication, more synthesis of antibiotic resistance, and therefore it led to a new discovery of synthesizing the desired biomolecules. Now, tools of recombinant DNA technology. What is required for a new DNA to be formed for the genetic engineering? So let's see here the restrictive enzymes, very important. Polymerase enzyme, ligases, vectors, host. All these are must for genetic engineering, for the recombinant DNA to be formed. Now, restrictive enzymes, scientists have been working on it. From where do they get these restrictive enzymes? Now, the enzymes are the catalysts. They work like catalysts because enzymes are catalysts. Scientists have taken out the enzyme from the bacteria itself. The bacteria which have those natural enzymes, those catalysts for defending themselves for certain reactions, they have those enzymes. The scientists have taken out these enzymes one after another. Now we have 900 restrictive enzymes taken out of 230 strains of bacteria. So slowly scientists are developing more and more restrictive enzymes. Restrictive enzyme, they restrict, they cut, they stop something, restricts. So therefore known as restrictive enzymes. So now in 1963 in Escherichia coli, when the bacteriophages they entered, they released restrictive enzymes. One which was used for giving a cut, and the other was used for added methyl group to the DNA. So the one which cuts is known as restrictive endonuclease. So but it was characterized, it was utilized only after 5 years. So now the first one you can say is Thankyou. Second, now these enzymes are taken from the bacteria Haemophilus influenzae. It has been taken from them. Now scientists, what they realize, that this restrictive enzymes, they recognize the six base pairs of DNA, and they recognize the sequence and they give a cut at a particular position. They analyze, they inspect the sequence, and then they give the cut. After giving the cut, there are sticky ends which are produced, which helps to take our DNA to the desired position. Now the cut has to be given on the other DNA also. So this is how it works. Our desired DNA can be transferred from this bacteria from wherever we want, but the cutting is possible only from the with the help of restrictive enzymes, which is taken from the bacteria. Now, let us see how this enzyme works. The enzyme cuts both DNA strands at the same site. So this is the vector DNA. You can see it is in the sequence GAATTC. GATTC, that is known as palindrome, that is read same way from this side as well, like the word Malayalam. You can spell Malayalam from the start as well as end. So same way you can read GAATTC, GAATTC. So that is the palindrome. Now this bacteria will give a cut in a fashion where just little bit away from the mid of the palindrome center, so that sticky ends, there are free strands which are given out, which forms the sticky ends, and that helps the DNA ligase to make it attached to the other DNA cut which we have given. So here, if this sequence you can see 5-3. Now this enzyme, restrictive enzymes, will give a cut in this way where it is GAATTC. Here the other DNA, how it has cut, here again it is GAATTC. So it is like a puzzle which is going to fix here. Both have sticky ends. So when you add DNA ligase, that will fix like a puzzle. So here this is the vector DNA, and this is the foreign DNA. Now you have to attach this desired DNA to the vector so that we get the desired genes for production of protein, hormone, enzymes, whatever we want. So here this is the foreign DNA, the desired DNA which we want, which has to be fixed to the vector. So this sequence have to be right 5-3. It is palindrome because they are same from both the sides. So here this is the foreign DNA, that also has to be cut in a particular fashion so that the sequence can be fixed one about the other. They both have sticky ends and the free ends, which helps the DNA ligase to fix in this manner. So DNA joins at sticky ends. Now this is the recombinant DNA which we get now, that has to be transferred in the host. Now this enzyme which we have used, the restrictive enzymes which we have used is E. coli. Now E is for Escherichia coli. Now R is for the strain, and I is for the Roman number. This is not I, this is number 1 in Roman 1. So this is the first Escherichia coli R enzyme. So this is how the names are given for the restrictive enzymes from the bacteria which they have been taken out. This is the genus, species, strain, and this is the Roman number 1, 2, 3, whatever is given. So this is number 1. It cuts the DNA with bases G and A only when the sequence GAATTC is present in the DNA. So these enzymes will inspect the sequence before cutting. They are enzymes, so they are catalysts. They have to inspect and then give a cut in a particular fashion so that both are cut by the same enzymes. Therefore they develop sticky ends. Once they develop sticky ends because of some sequence hanging, and then they stick by the DNA ligases. So now we have the recombinant DNA with the help of this enzymes E. coli. I, that is 1, that is 1. Cuts. Now this is how this enzyme works in forming a recombinant DNA. So now we have 900 enzymes from the 230 strains. Now how does the restrictive endonuclease functions? First, it inspects the length of the DNA sequence. It inspects, sees how much is the length of the sequence. It recognizes the specific sequence, whether they want to cut at this function, this recognition site, whether they have to cut at that particular point. So they recognize that specific sequence, which is specific recognition sequence. Once it has found that sequence, then it binds to the DNA. It binds to the DNA. Then it cuts each of the strands of the double helix at specific points. After binding with the DNA, they give a cut at the specific point. Now, specific point where at the on the DNA which has got the sugar phosphate backbone. So the backbone is there, sugar phosphate backbone is there, but what they are cutting? The nucleotide. They are cutting at a specific point. So this is how the restrictive endonuclease enzymes they work. Now, this is the diagrammatic representation of recombinant technology. So, here's a foreign DNA, the DNA which has got a specific function of producing protein, hormone, whatever. We want this DNA, foreign DNA, we want this some part which has to be multiplied. So, what we are going to give a cut to this DNA with the restrictive enzyme. So same restriction enzyme cutting both foreign DNA and vector DNA at specific point. So here this is the foreign DNA, this is our plasmid, this is our vector which is going to transfer those special DNA, special genes in the host. So this is the vector, this is our DNA which we wanted to multiply. So we have to transfer in this vector. So this vector is a plasmid. Plasmid which is just a carrier. Vector means it is a carrier. When mosquito bites you and transfers the malarial parasite, it's a vector, just transfers the malarial parasite. So plasmid is also just a vector, it is a carrier. So a cut has to be given to the plasmid so that we can fix it. Now same enzyme has to be used because that enzyme will produce sticky ends when the same enzyme cuts the foreign DNA as well as the plasmid. So same enzyme has to be produced. Now it has already cut the DNA here and now it is cutting the plasmid here. Now when it cuts the plasmid, there is a space, it has removed some nucleotides and here there is a space. Now this is DNA. This DNA is transferred here in this space. Now this is a recombinant DNA, a new DNA which has to be multiplied. But how it is not going to multiply by itself? It has to be introduced in a host. It has to utilize the enzymes of the host for multiplication, for polymerase chain reaction. So here, once the recombinant DNA has been formed with the plasmid is introduced in Escherichia coli. That's the microorganism, that's the bacteria. Now this recombinant gene, now all this is shown in big structure, but now it is a small structure which has entered the cell. It has entered the cell. This structure has entered the cell. So now once it has entered the cell, the host cell is going to multiply. When it multiplies, this plasmid will also multiply. Now this plasmid consists of DNA, which is going to produce either insulin, protein, whatever is required, that also gets multiplied. Supposing it is for insulin, as the bacteria they go on multiplying, the insulin in it also multiplies. Now once the insulin is multiplied, it is extracted from the bacteria, put into bottles. Now you have insulin which can be injected or utilized by the humans. So this is how this recombinant DNA helps the human. It can be done for various purposes, for protein growth hormones or many more things. So this is how the technology helps us. This is how the recombinant DNA helps to multiply and be beneficial for the humans. Now, separation and isolation of DNA fragments. You have got the DNA fragment with the help of restrictive endonuclease. So, cutting off DNA with the help of restriction endonucleases. What you get is fragments of DNA. You have got the fragments. Now you have to separate those fragments which can be put into the vector and utilized. So what it is done by gel electrophoresis. Gel agarose is the gel which is put in the tray, and this DNA, it spreads in the gel and then it can be separated. How? Let's see. So gel electrophoresis is a process by which electric current is passed to the matrix. It is negatively charged. This DNA is negatively charged. So when the current is passed, they get attracted towards anode. They are anions. They get attracted towards anode, and therefore it is easy to separate. So you can see this tray which is filled with the agarose. It's a matrix. It is derived from algae. The cellulose, the polysaccharide is derived from the algae, that is utilized as gel, that's utilized as matrix. So here you have the matrix. It's the natural polysaccharide. It is natural substance which helps in spreading giving this DNA spread out. So here is the matrix which is made up of agarose. Here there are wells. There are some small tiny space where you put the DNA. Now this DNA is going to travel by the sieving effect. There are minute space in between the gel. So by sieving effect, they will just move forwards. Now on this matrix, the current is passed. So here the DNA will get attracted towards anode and it will start traveling fast. It will without current it will go very slowly, but with the current it will travel very fast. So now it has spread out with great speed. You have DNA here in agarose gel, but you cannot see. What you will do is you will dye it. You will color it, you will stain it. So which is the stain which is used? It is ethyl bromide. It is ethyl bromide which is the stain. It has to be stained with ethyl bromide. And then even then you won't be able to see unless and until you see under UV rays. And once it is stained with ethyl bromide and you see with the UV rays, behold, you see the orange color. You can see this orange color, that is DNA. So in this matrix in agarose, DNA is spread and orange color is the DNA. So now what, like jelly, this looks like jelly. So you just cut out this orange structure and you get the DNA. Now this cutting, spreading and cutting of this DNA from this matrix, this solvent, is known as elution. Elution. The bands, orange color bands which you can see you have cut out, that process is known as elution. So what is elution? Bands are cut out. So here you have a small tray, a typical agarose gel electrophoresis showing migration of undigested and digested set of DNA fragments. Lane 1 is undigested. Lane 2nd and 4th, they are digested. So you can see how the DNA spreads. We want DNA so they can be put into vector and utilized. So this is how separation of DNA takes place by this electrophoresis. That means you are utilizing the electric current for separation of DNA. DNA is negatively charged. It gets attracted towards a node and gets attracted, therefore it travels at a faster speed and it spreads out in the matrix, in the gel, in the agarose. So here you have the spread out DNA. So here cutting of DNA by restriction endonuclease, fragments of DNA, they will be separated by gel electrophoresis here and they get attracted towards a node, they are negatively charged. DNA gets separate according to size by sieving. Sieving is the small particles will move wherever they find the space. So that is sieving effect. They are stained with ethyl bromide. They are stain is put, ethyl bromide is put on it. And when you put UV light, you get orange bands. And these bands are cut out. That process is known as elution. So now this is constructing recombinant DNA by joining them with cloning vector. So now this band, orange band, has got DNA. This DNA has to be reconstructed. They have to be put into the vector so that the cloning takes place. So you are constructing recombinant DNA by joining. This is recombinant DNA which you are getting. So now constructing recombinant DNA by joining them with cloning vector. So after this process, the cloning of vector will take place. This DNA will be transferred into vector and vector will clone this DNA. So this is how separation and isolation of DNA fragment takes place with the help of gel electrophoresis process. Now the cloning vectors. Cloning vectors are engineered in such a way that the foreign DNA gets linked to it. They are linked to the foreign DNA, the alien DNA. And they can select the recombinant from the non-recombinant. So these vectors which are going to transfer our DNA which we have selected, they are just going to transfer by combining with the alien DNA, by linking with the foreign DNA. So here our vectors, our heroes are plasmid and bacteriophage. Now plasmid are the extra genetic material of the bacteria. Bacteriophage are the viruses which infects the bacteria. Now they are used as vectors for transferring our DNA which we need to transfer and then multiply. So these are going to replicate within the bacterial cell. Plasmids are present in bacterial cells. They are going to multiply in the bacterial cells. They are independent of the control of chromosomal DNA. If plasmid is removed from bacteria, bacteria still will be able to multiply. So they are extra structure, genetic structures which are the plasmid. So they are independent of the control of chromosomal DNA. Bacteriophage has got very high number of genome within the bacterial cells. They will multiply at very fast rate. So they are going to create many copies. Now plasmid may make 1 or 2 copies, or it may make 15 to 100 copies per cell. So these are the vectors which bacteriophage very high. Here it is little bit slow. It might be 1 to 2 copies per cell or 15 to 100 copies per cell. So here they are going to accept the alien DNA, piece of DNA. This vectors are plasmid and bacteriophage. They are going to accept the alien piece of DNA. Now vectors are engineered for easy linking of foreign DNA. Foreign DNA has to get attached to this vector. They are capable of selection of recombinant from the non-recombinant. Recombinant, we want it to multiply. So vectors are going to select the recombinant. Non-recombinant, we have not made non-recombinant. So it is going to discard. So selection of the recombinant DNA is done by the vectors. So this is how the cloning by vectors takes place.